Dear students, I do not teach life science. I teach the science of life. Today's topic is heredity. In reality, heredity is the science of life. Follow, whenever a child is born, the family members start comparing of his appearance to whom he or she resembles most, father or mother. So, the characteristics features of father and mother are transferred to the offspring. It is heredity. So, the process of transmission of parental characters from parents to offsprings through reproduction is called heredity. So how does it occur? You know, by the union of a male, that is haploid, and a female gamete, a diploid zygote is formed. It is a zygote, diploid zygote is formed. So the chromosomes, haploid sets of chromosome form the male and female gamete, that is father and mother body, assemble together within the zygote to form the diploid. This zygote repeatedly divides by mitosis to form an embryo and ultimately to an offspring. Since from zygote offspring is produced, so we can say the chromosomes of the zygote, or more precisely the chromosomes of the father and mother, control the characteristic features of the offspring and its expression. That is why there are many similarities are seen among the members of the same family, like height, shape of face, shape of hair, teeth, earlobe, etc. Now you say that this transmission, how does it occur? The first scientific explanation was given by an abbot or priest of Austria known as Gregor Johann Mendel. Gregor Johann Mendel. Mendel was not a scientist. In his time, chromosomes and genes were not discovered. Without any idea about the chromosome and gene, his such epoch-making discovery is really amazing. But it is a matter of regret that he did not get any honor or recognition during his lifetime for his discovery. After his death, after the rediscovery of his findings, it is observed he discovered the basic principles of heredity. From then, he is regarded as the father of genetics. The genetics, what is genetics? The branch of biology which deals with the study of gene, its structure, transmission, etc. is known as genetics. Now you see that there is the word that like begets like. It means the offsprings resemble the parents. That is why from a mango plant, from a dog, from a man, born. But these offsprings do not resemble exactly like their parents. That is, even the two offsprings of a parent are not alike. So these differences are known as variations, you know, already studied. So what are the causes of variations? The number one the causes of variation crossing over. Crossing over. You have already studied it in meiosis. During meiosis, crossing over takes place. As a result, rearrangement of gene occurs and the offsprings produced by the union of gamete, new characters appear. That is the variation. Number 2, mutation. Any sudden but permanent change in the germ plasm is known as germ plasm, which leads to the appearance of new character, is known as mutation. Due to mutation, the changes occur in the shape, number of chromosome, change in the DNA gene occurs, so new characters will appear. The shape of chromosome changes means, suppose it is a chromosome. Here the genes are present A, B, C, D, etc. By any means, any part of this chromosome gets detached and ultimately removed from it, and then the shape changes. Similarly, number also changes. For example, in human body there are 46 number of chromosomes in each cell. It may be 47, 45, etc. Number 3, random arrangement of chromosomes. Arrangement of chromosomes. You have already studied in meiosis, that is during metaphase 1 of meiosis, the homologous chromosomes are arranged equidistant from the equator. It is the equator, that is one is placed towards one pole, and its homologous pair in the opposite pole, that is 1, 1, 2, 2, and so on. And these two homologous chromosomes, one from father body, that is paternal, and other from mother body, that is maternal. But all the paternal chromosomes will arrange in one side and another, that is all the maternal towards the opposite side, never occurs. And as they randomly arrange and ultimately the gametes are formed, and also by the union of these gametes, the offsprings produce, new characters appear. Now, you see the chromosomes bear the characteristic features. More precisely, the genes bear the characteristic features. They are the carriers. In the evolutionary stage, the first organisms appeared in this living world were haploid. That is, there in each cell haploid number of chromosomes were present. Till now you see the haploid organisms in this living world like the Spirogyra, Mucor, etc. So in a haploid cell there is the one set of chromosomes, and within these chromosomes the genes are present. The single gene controls a single character. But in course of evolution, the diploid organisms appear, and in diploid organisms the chromosomes become double, that is homologous, 1, 1, 2, 2, and so on. And since the homologous chromosomes are identical, that is from one chromosome another chromosomes appear by mutation. And since they are identical, means they are shape, their genetic characters same. So the genes present in the previous chromosome, the similar type of gene appear also in its counterpart, and a pair of genes thus form which control a character. In this way, the two genes control a character in diploid organisms. Suppose in human body also, a pair of genes control the characters, like the color of eye, brown and blue, the shape of hair, the curly hair, straight hair, earlobe, free earlobe or attached earlobe, etc. And in this way, these organisms, diploid organisms which evolve now more advanced type than the haploid one. Now you see I explain you some terms related to heredity, and these terms are very important. Number one, character. So any inheritable feature of an organism is known as character. Any inheritable feature of an organism is called character. Number two, trait. The character which is inherited in a particular form, that is, in which form a character is inherited is the trait. Generally, each character has two alternate features, that is, traits. For example, in Mendel's experiment, the height character, the length of stem, is two alternate forms, tall and dwarf. So, tall and dwarf are the traits. Height is the character, that is the expression of the character either in the form of tall or in the form of dwarf. It is the trait. For almost all characters have two alternate traits. For example, in human body, that is the color of eye, the brown and blue, the shape of the hair, the curly and straight, which I say before. But these two alternate traits are formed from a gene which is regarded as Y gene. Suppose it is the chromosome, the first chromosome 1, and from which its counter trait part 1 homologous pair appear. The gene which was present in the first chromosome is regarded as Y gene, and from this Y gene, by mutation, the new gene appear. This new gene also regarded as a mutated gene, and in this way alternate form. Almost all characters have two alternate, but mutation, if occurs more than once of a gene, then more than two alternate forms appear. For example, in human blood group, that is human blood group ABO is controlled by three genes, I, IA and IB. I is the Y gene from which by mutation, two types of mutation, IA and IB. Although in a population more than two genes are present, but in a given individual at a time only two genes are present, because the homologous chromosomes are two in number. Here in man, suppose either I or IA, and in such a patient. Okay, number 3, locus. Locus. A specific gene which is present in a particular position of a particular chromosome. This position is called locus of that gene. That is a chromosome. Here in particular position a gene is located. This position is called locus. For example, in your classroom the low bench is the chromosome and you are the genes, and so the position where you sit, that is this position is regarded as the locus of you. Then allele or allelomorph. Allelomorph. देखो, a pair of homologous chromosomes, the two genes situated in the same locus of a homologous chromosome pair are said to be allele to each other. In reality, allele is the each of two or more alternate forms of a gene that appear due to mutation. Here, suppose the height character of the pea plant, its alternate form tall and dwarf. Tall and dwarf. So tall and dwarf, the alternate form, these are the alleles. But in a homologous chromosome pair, the genes which are present may be in similar form and alternate form. That is, in homologous pair, in this position there is the tall gene, and in this position there is the tall gene. It may also happen, and there may be tall gene and dwarf gene. So both are the alleles. The first one is the homozygous allele and the second one is the heterozygous allele. So what is the main difference between the allele and gene? The gene is the specific form or structure, that is functional segment of DNA which specifies a particular trait or particular feature, and allele is the particular form of a gene, that is in which form a gene is expressed, that is the allele. These allele may be present in one or more forms. Number 4. Homozygous and heterozygous. Homozygous and heterozygous. A pair of genes situated on the same locus of a homologous chromosome pair are said to be allele to each other, you say. So you also say a pair of allelic gene, we can say a pair of allelic gene responsible for a character bearing the similar trait. That is, here the tall, here the tall, here the dwarf, here the dwarf. Then this character or the organism for that character is regarded as homozygous or heterozygous. A pair of allelic gene responsible for a character bearing the contrasting trait. Suppose here tall and here dwarf. Then this character or the organism for that character is known as heterozygous. One organism cannot be homozygous for all characters or heterozygous for all characters. That is, for one character he may be homozygous and for another character he may be heterozygous. Phenotype and genotype. Phenotype, externally manifested character of an organism is the phenotype, that which we see from the external feature, it is the phenotype. But genetic combination of the character is the genotype. For example, you say the tall, dwarf, black, white, these terms are phenotype. But for tall, which genes are present in the allelic pair? Suppose in a homologous chromosome pair, there is a tall gene and another the allelic gene, there is the tall gene, and it is known as the homozygous tall or pure. And if there is the capital T, that is tall, and another dwarf, here also tall, it is the heterozygous. So homozygous, heterozygous, these terms are regarded as genotype. Tall, dwarf, these terms are regarded as phenotype. Then dominant and recessive. The trait which is expressed in both homozygous and heterozygous condition is known as dominant, and that trait which is not expressed in heterozygous condition and remain suppressed condition is called recessive. So, dominant trait is expressed in both homozygous and heterozygous condition. Here you see that is, it is the two homologous chromosome. Here the one character, the tall, another character, tall, it is homozygous. So the plant should be tall, and here the one chromosome it bears the tall gene and another chromosome bears the dwarf gene. It is also tall. There is homozygous and heterozygous condition. The trait is expressed dominant. The tall is the dominant, but dwarf is remains suppressed condition in heterozygous condition, and to be expressed it must be homozygous. So it is the recessive. Next, unit of heredity. Unit of heredity is the gene, but Mendel did not know the gene and he simply termed it as factor. So factor is also the unit of heredity. Hybridization. Hybridization. When a cross is done or a sexual reproduction is done between the two organisms of the same species, considering the characters with the contrasting traits, then it is known as hybridization, and the offsprings produced known as the hybrid. For example, if the tall plant and dwarf plant is crossed, that is contrasting trait tall and dwarf of the two organisms, and then the offsprings produced both the genes, tall gene and dwarf gene, it is the hybrid, and this process is the hybridization. Then monohybrid cause, dihybrid cause. These are the Mendel's experiments. Mendel experimented with one character, single character at a time or two characters at a time. When a cross is done between two characters with their contrasting trait, it is dihybrid. And when one character with its contrasting traits, then it is known as monohybrid. Parental generation. Parental generation. Parental generation is the generation from which a cross starts, and it is denoted by P. Generally, parental generation starts with pure line variety, that is both the pure tall, both the pure dwarf. First filial generation. First filial generation, or F1, is denoted by F1. The offsprings produced by the cross between the organisms of the parental generation, known as the first filial generation, F1. Similarly, from the F1 generation, the F2 generations are formed. There is second filial generation. Now we complete the different terms. Now we come the Mendel's work. What Mendel did? Number one, selection of parent. Mendel selected sweet pea plant, Pisum sativum. It is the parent, it from the parental generation. Then pure line variety. After the selection of parents, he became sure the taken plant were pure or homozygous. For this, he took the particular plant, suppose tall plant, and the dwarf plant, because we know the cross will be done between the two contrasting, and he allowed for self-pollination among the tall plants for generations. When he observed that tall plants generation after generation produce only the tall plants, he became sure it is pure. And similar phenomenon he done in case of dwarf plant. It is the pure line variety. Number 3, cross pollination and fertilization. He cultivated the pure tall and pure dwarf plant, and then in bud stage he opened the petals of the dwarf flowers of dwarf plant very carefully and removed the male sex organs, and that is turned them into female flower. It is known as emasculation, that is surgical removal of male sex organs from a bisexual flower to make it a unisexual, that is female flower. It is emasculation. After that, he covered the flowers with the help of net. And after maturity, he collected the pollen grains from the tall plants and with the help of brush and spread them over the stigma of the dwarf flowers. And ultimately, the cross-pollination and fertilization occur. And then he covered these flowers with the help of net. And after formation of fruits and seeds, he collected them and ultimately he cultivated them to get the result. Now, reasons behind Mendel's success. The Mendel selected the sweet pea plant. It has the great advantage. Pea plant is a bisexual, that is, flowers are bisexual, self-pollination is the rule that occurs easily, but cross-pollination be easily done. There are so many characters with definite contrasting traits and which are distinctly dominant and recessive. The seeds produced huge number, and that gave him the facilitation of the ratio. The hybrid offsprings are fertile. Generally hybrids are sterile, but hybrid offsprings are fertile, so he could experiment for F2, F3 generations. He simply used the term factor without any complexity, and he only selected 1 or 2 characters at a time, and he selected the plants only which are distinctly dominant and recessive. Besides, the pea plant can be grown easily within a short area. These are the some of the causes of the Mendel's success. Now, Mendel selected 7 characters with 7 pairs of contrasting traits, which are like length of stem, the dominant is the tall and recessive is the dwarf. In this way, the 7 characters and their contrasting traits are like the shape of seed, the round and wrinkled, color of cotyledon, yellow and green, the shape of the pod, smooth or inverted and wrinkled, the color of the pod green, yellow, position of flower, axillary and apical, color of flower, the violet, that is purple and white, etc. Now we come the Mendel's experiment. The Mendel's experiment, that is first monohybrid cause and then dihybrid cause. The monohybrid cause, that is when a cross is done between the two organisms of the same species considering one character with its contrasting trait. It is known as monohybrid cause. The selected plant the sweet pea plant, selected character, that is one character, the length of stem of pea plant, contrasting traits, tall and dwarf. Tall is the dominant and dwarf, it is the recessive. That is, this capital letter, small letter, generally the tall, the first letter of it is taken as the dominant, when it is dominant, and the dwarf and its small letter is taken. You can write the small d. It is the general rule, the capital letter represents the dominant and small letter represents the recessive. Then genotype of pure tall, genotype of pure dwarf. Here we take the 2 capital T, 2 small t. That means in a homologous chromosome pair, the 2 genes are present which are similar, that is capital T, capital T. Similarly, small t, and that is why we take the 2 letters. Now he crossed between the pure tall and pure dwarf. The pure tall, it is a male, symbol of male, and pure dwarf, symbol female, parental generation, both pure. Capital T, small t, these are the gametes. Here the 1 or 2 gametes not formed, it is a type only, and here the gametes that are formed all the similar types, so you can write only one. And hence here we write the capital T and small t, that is meiosis and segregation. Then cross fertilization, and by cross fertilization the offsprings produced both via the capital T and small t, and he collected the seed, and after cultivation he obtained all tall plants. It is the F1, first filial generation, and as the F1 generation all the plants were tall, so he allowed self-pollination. Capital T, small t, capital T, small t, and these are not the 2 plants, it is the single flower, and in single flower the male sex organ and female sex organ, since here self-pollination occurs. And so here 2 types of gametes form, capital T, small t type and capital T, small t type gametes. And any one type of male gamete can fertilize any other type of female gamete. If capital T fertilizes the capital T, then capital T. In this way, capital T, small t, capital T, small t, small t, small t. F2. From the F2 generation, he obtained the two types of plant in phenotype, 3 part tall and 1 part dwarf. So phenotypic ratio is the tall is to dwarf. Phenotypic ratio tall is to dwarf equal to 3:1. But you see here genotype, the 25% both the capital T, so it is homozygous, and 50% capital T small t, so the heterozygous, and 25% small t, so homozygous. So genotypic ratio homozygous tall is to heterozygous tall is to homozygous dwarf, 1:2:1. And this cross you can write with the help of checkerboard, that is Punnett square. Scientist Punnett first discovered it, and for this you do the gametes, that is male, female, capital T, small t, capital T, small t, and now you put them into the checkerboard. And as the 2 types of male gamete, 2 types of female gamete, so only the 4 types of offsprings, the male, female. Here you put the male gametes, here put the female gamete, and then capital T, small t, F2, and then you write the phenotypic ratio and genotypic ratio. It is the monohybrid cross. After the performing the monohybrid cross, he formulated his first law, the law of segregation. The factors, according to this law, the factors do not blend in heterozygous condition. He said the factor, now you know the gene. Factors, that is bracketed within bracket the gene. Factors do not blend in heterozygous condition. During gametogenesis, that is formation of gamete, they are segregated, that is differentiated, and are distributed into different gametes. Mendel, by cross between pure tall and pure dwarf, he obtained all tall plants. So he was astonished what was the fate of the dwarf or recessive. Is it blended with the dominant, that is tall, or it is lost? And for this he allowed the self-pollination. And in F2 he again obtained the dwarf. And then he formulated the first law, that is the genes or factors do not blend in heterozygous condition, and during gametogenesis they are segregated and are distributed in different gametes. And from these Mendel's experiment we also can see why did Mendel obtain all tall plants in F1? Because Mendel crossed with the pure variety, that is tall we know the dominant, dwarf we know the recessive. The tall only produce the tall gamete and dwarf only produce the dwarf gametes, and by cross between the heterozygous condition, tall and dwarf allele come side by side. Since the tall is the dominant, so it is expressed in phenotype. Okay.